In this lecture, we will discuss about protein structure and function, the basics of protein building blocks and how they form the sequence and the structures and what are the major functions of proteins. Just to refresh ourselves, what did we discuss in the previous class? Correct different databases. So, correct, right? What is database? It is a collection of data in the computable organized form, right? Fine. So, we discussed about databases and what are the characteristic features of databases and what are the factors one has to consider for the development of a database and so on. Then we discussed about relational databases, right, and the collection of databases. Where shall we get the collection of databases? In nucleic acid research. This is they publish every year, right, and also they maintain a website to give the collection of all the databases available in the literature. As well as you can see in the PubMed, as well as another journal called Database, they also publish quite a number of articles about database. Then we moved on to the databases related with nucleic acids. First we did about the nucleic acid sequence databases, right. What are databases dealt with nucleic acid sequences? DDBJ, EMBL and GenBank, right, and then we discussed about the properties, right. So, what are the dinucleotide properties? We discussed about nuclear property database. Then we discussed about the literature databases, right. What is the commonly used literature databases for biological sciences? PubMed, right. So, you can extensively use PubMed to obtain the information regarding the literature published in biological sciences. So, if you look into these biological macromolecules, right, they are classified into three major groups. What are the three major biological macromolecules? Proteins, nucleic acids, carbohydrates, right. So, which is nucleic acids we discussed in the previous classes, right. These ones want to carry genetic information, right, and we talk about carbohydrates, right. If it is a staple of human diet, in the form of glucose or fructose, right, then also it is structural protective elements, right, depending upon the linkage, right, cellulose and so on. Then consider the proteins, this is extremely important, it is extremely versatile in their functions in all living organisms. Proteins perform diverse functions in living organisms. If you talk about the proteins, what is the building block of proteins? Right, it is amino acids, right. So, we have the amino acids, right, it is the building block of proteins. Why it is called amino acids? Because it contains an amine group, NH2 group and carboxyl group, right, and this is the side chain. So, it varies. So, because of the amine group and the carboxyl group, right, we have the amino acids. So, first I like to see about the types of amino acids, right. There are 20 naturally occurring amino acids, right. So, here I give you the list of the 20 amino acid residues. In the later slides we use the 3-letter codes or the 1-letter code. So, here I give you a table regarding all the 20 amino acid residues along with their 3-letter and 1-letter codes. So, we see alanine, so this is ALA and just A. Likewise, for several amino acid residues you can use the first letter of each amino acid, but some cases if you see alanine and aspartic acid both are start with A. So, preferably they put for alanine they give A. So, on the aspartic acid they pronounce as aspartic acid, so they put D. Likewise, they assign 20 letters for the 20 amino acid residues, right. If you see there are the letters which are not used in the amino acid residues. So, 20 different amino acid residues, they are majorly classified into two different groups, right, based on their characteristics, right. So, two major groups are hydrophobic residues and hydrophilic residues, right. What is phobic? They dislike, right. So, this hydro means water. So, amino acid residues which hate the water, right. So, mainly they are containing the aliphatic groups, they are hydrophobic residues, and the residues which have high tendency to interact, so they are hydrophilic residues. For simplicity, I classified 10 amino acids into hydrophobic and another 10 into hydrophilic. So, in the hydrophobic amino acid residues, I made into 4 classifications. So, first one is a glycine. So, because it has no side chain, right, because N, C alpha, C, right, they are mentioned atoms, and the side chain is a H. This is the only one amino acid which are not chiral, right, because here all the 4s are not different groups, but all the 19 amino acid residues the groups attached to the carbon are different, so are chiral amino acids. So, the second group aliphatic amino acid residues, right. What is aliphatic amino acids? Right, alkyl group, right, that is CH2 group. So, if you see all the amino acid residues like alanine or leucine or isoleucine or valine they contain CH2 groups. So, they are called aliphatic amino acids. Then here are 3 cases I put, right. So, this contains the rings, right. So, they are called aromatic amino acid residues, right. What are 3 aromatic amino acids? Phenylalanine, tyrosine and tryptophan. So, here I put another group sulfur containing amino acids. There are 2 amino acid residues contain sulfur, right, one is methionine and one is cysteine. Among the two, one will form disulfide bonds. Which will form disulfide bonds? Cysteine, right. So, cysteine will form the disulfide bonds, right. So, mainly classified two groups, and among the two groups hydrophobic residues we classify into 4 different classes: glycine, aliphatic, aromatic and sulfur containing. Likewise, if you look into the hydrophilic residues, here I classify into 3 different groups. One is negative charge, and because it contains the acid group, so aspartic acid and glutamic acid, and positive charge lysine, arginine and histidine, right, and others are polar residues. Polar residues are asparagine, glutamine, serine, threonine and proline. So, majorly you classify 2 groups, and among the 2 groups you classify subgroups, right. These groups are important to understand specifically among the mutations. Mutations represent the replacement of amino acid residues. To understand the protein structure or function, it is important to understand, to know about the characteristic features of each amino acid. Now, what are the various chemical groups involved in 20 amino acid residues? There are various chemical groups such as alcohol. Which amino acid contains alcohol? Serine, right. Serine contains alcohol. So, which one has the acid? Aspartic acid, glutamic acid, right. So, always you can have the amine, lysine, arginine, second is amine group. Likewise, aliphatic, alanine, valine contain aliphatic, and the aromatic, tryptophan, tyrosine, right. So, they contain several chemical groups, right. With these chemical groups are important to understand the structure and function of proteins, because these groups initiate some process for folding, and they try to provide the stability of the system of the proteins. So, what are the various properties of amino acids? With 20 amino acid residues classified in 2 different groups, as well as they belong to different chemical groups. Due to the chemical groups, they form different types of interactions. First one is they take the non-polar residues or the hydrophobic residues, right. So, mainly with alkyl groups or aromatic groups, right. So, these residues tend to form the hydrophobic interactions. What is hydrophobic interaction? Form a core, right. The tendency of these non-polar groups to adhere to one another, come close to one another in aqueous environment. So, if you see this aqueous environment, right. So, these groups they come close to each other, they form a common group, like oil water experiment. If you put an oil in water, right, the oil they tends to come close to each other, right. So, they form a group. So, these are called hydrophobic interactions. Likewise, the polar amino acid residues like the hydrophilic residues, they are the different types of residue functional groups such as acids or amides or alcohols and amines. So, when you look into the polarity of these chemical groups, okay, here I give the amino acid residues with increased order of polarity, amides and acid, then alcohol, and amine is more than ether and alkane. So, there are 3 different types of amino acids. So, some of them are neutral like glutamine and asparagine, and acidic like glutamic acid, aspartic acid, and some are basic lysine and arginine. So, if these residues they have a tendency to form various types of interactions. For example, if you take the acidic and basic amino acids, they tend to form which type of interactions? Salt bridge and electrostatic interactions, because the interaction between the positive charge residues and the negative charge residues. Likewise, the other polar residues they prefer to form hydrogen bonds, right. Likewise, the 20 amino acid residues they have the tendency to form different types of interactions to initiate folding and to maintain the stability of the proteins. Eventually, that is responsible for the function. So, now I have a question. So, I have 4 amino acids here, right, the leucine, phenylalanine, valine and alanine. So, could you rank these amino acids to increasing non-polarity? That means starting with the lowest to the highest, considering aromatic is higher non-polarity. So, which one is the lowest one? Alanine followed by valine, leucine, phenylalanine. Why this alanine is less non-polar than valine? Small, right. Alanine contains how many CH2 groups? Only one, you can see here. So, this alanine, this one CH2 group. Valine 1, 2, 3, right. So, leucine 1, 2, 3, 4, right. So, accordingly, if you look into the number of CH2 groups, alanine has less, valine is more. So, valine is more hydrophobic than alanine, right. You can increase the order of non-polarity. Then the second question, I have 5, 6 amino acids: serine, glutamic acid, aspartic acid, lysine, alanine and glutamic acid. So, can you rank these amino acids based on increasing polarity? That means one is more non-polar, alanine, because this one is the aliphatic one. So, this is highly non-polar. So, then the others, okay, this is the order. First amide has more, then acidic acid, and then alcohol, then amine, and then ether and alkane. So, we compare these chemical groups, right, these are the chemical groups involved in the 20 different amino acid residues here, right. So, you can see this alanine is nonpolar and then this amine lysine and serine, this alcohol, and these two are acid, right. So, among these two, glutamic acid and aspartic acid, this has one CH group more. So, glutamic acid, then aspartic acid, and finally the glutamine. So, now the third question. So, if you have the amino acids isoleucine or alanine, which one is more insoluble? Isoleucine, right, because it has more CH groups, it is more insoluble than alanine. So, which amino acid is most soluble in water, lysine or serine? Serine, right, because alcohol group is more soluble in water. So, if you have 20 different amino acid residues, they have their own characteristic features. Due to these characteristic features, right, they tend to interact with each other, right, in protein structures. So, now how they form a protein chain with 20 different amino acid residues, right, they are the building blocks of proteins, and how they form a protein chain. So, here you put the chain 1, this is the amino acid 1, right, NH3COOH, and here this is the side chain, I put R1, and this is hydrogen. So, this is the second one, then they combine together, right. So, by the elimination of water molecules, right, this OH and the H, they form water, the elimination of the water, then they form the peptide bond, right. So, if you see now CO is here and this NH is here, right. So, this form the strong bond, this is a partial double bond character, right. So, this bond is called peptide bond, that is NH CO bond is the peptide bond, right. This is the residue 1, this is residue 2, right, and this is a peptide bond. Likewise, you combine more and more amino acid residues and finally, they form the protein chain, right. So, if you see many peptides they form together, they form a polypeptides, right, but and then finally, they form the functional protein. For a functional protein, so the order of this amino acid is specific, nature selects a specific combination of amino acid residues to form a functional protein, right. For example, if you have polypeptide, the question is all polypeptides are proteins or not? No, but all proteins are polypeptides or not? Yes, right, all proteins are polypeptides because you can say polypeptide, but polypeptides are not proteins because you can form polypeptides, but they are not functional, right, because nature selects a particular combination of amino acids. Now, if you combine different amino acid residues, right, a specific combination produces a functional protein, right, and if you take the proteins, there are different types. Here you mainly classify into 3 different groups, one is the globular proteins, fibrous proteins and membrane proteins, right. Globular proteins are generally globular in shape and they are mainly functional proteins, they perform various functions, right. And if you go the fibrous proteins, right, it is mainly structural proteins, also called as structural proteins, it gives the strength to the system, right. For example, where can we find the fibrous proteins? Nails, tendons and the hair and so on, right, it is mainly important in the structural role. The other type of proteins, they are called membrane proteins, right. Membrane proteins are one, the proteins are embedded in the lipid environment in the membrane region. So, here I show the examples, here this is an example for the globular proteins, right, this is an example for a fibrous protein, right, you can see elongated chains, and this is an example for a membrane proteins. When looking at membrane proteins, they are of 2 types, here one is alpha helical membrane proteins and another is beta barrel membrane proteins. This is schematic diagram, right, if you see the gram negative bacteria, so you can see a cytoplasm and they have the periplasm here in outer space. Here there are 2 membranes, one is the inner membrane, right, inner membrane is the one which is between the cytoplasm and the periplasm, and the outer membrane is between periplasm and outer space, right. So, if you look into these membranes and the structures of the proteins which are accommodated within this membrane region, if you in the inner membrane, it is mainly the proteins in the inner membrane, right, they have alpha helical conformation, right, because mainly the functional point of view. So, they have the alpha helical conformation, and if you see the outer membrane, that is between periplasm and outer space, they are mainly with the beta sheets. So, these proteins are also called beta barrel membrane proteins, they are called inner membrane proteins or alpha helical membrane proteins, right, and here they are called the beta barrel membrane proteins, right. There is just one exception which having the different types of conformation, other than that almost all the membrane proteins which are known in the literature, they are having alpha helical membrane proteins in the inner membrane and beta barrel conformation in the outer membrane. So, now if you look into the globular proteins or fibrous proteins and membrane proteins, they have different structural and functional roles. I will explain a bit more about the functions of this protein. So, one of the most important functions of globular proteins, enzymes, enzymatic activity. So, there is the major function of these proteins. So, enzymes are specialized proteins with the catalytic activity that increase the rate of a chemical reaction. So, we take a full protein, right, only a few residues, the portion of this enzyme, say 3 to 4 amino acid residues, right, this is directly involved in the catalysis, right. So, the region which contain these catalytic residues, right, this will bind the substrate, right, so bind the substrate and then it carries out the reaction. There is another, this is called the active site, the residues, right, which are involved in the catalysis, these residues are called the active site residues. So, if you have one example, right, one is the carbonic anhydrase, right, this is a family of enzymes. This catalyze the interconversion of carbon dioxide and water to bicarbonate. So, we use carbon dioxide plus water and these enzymes they convert into bicarbonate and in tissues and high carbon dioxide concentration. In the case of lungs and low carbon dioxide concentration, this bicarbonate, right, this is reduced to CO2 plus H2O. Here is another example, here I show the example for the metal enzymes, like the active site of the most of the carbonic anhydrases, right, here you can see the contains the zinc ions, okay, the zinc ion. This is coordinated by different 3 histidines, you can see one histidine here, one histidine here, one histidine here. So, 3 histidines. So, these proteins are also called metal enzymes because they are coordinated with this zinc ion, right. So, then in enzymatic reactions, the molecules at the beginning of the process are called substrate, okay, here you can see this is the molecules which are beginning of the process, they are called substrates here, right, and this is the protein, right. So, this is the protein, so these are the substrates, right, and the one which is converted to the different molecules they are called products. So, you can see the products here, right. So, if you see the enzyme protein here, here the substrates they bind with the proteins at the active site, okay, here this is the active sites. I mentioned that a few residues, 3 to 4 residues which is involved in the catalysis and this site is called active site, right. So, here it interacts with these active sites and then enzyme changes the shape and it binds the substrate and then this when they produce the groups that the products are coming out. So, do the rate of the reaction, they increase the reaction and then finally the product is coming out, right. So, if you see here this is the substrate and the protein, right, they interact and then finally have the products and the products are coming out. And then different types of enzymes, for example, one is the alcohol dehydrogenase, this is the enzyme, it facilitates to break down the alcohol because alcohol is toxic, right, into the others like aldehydes or ketones, right. So, if you want to consume alcohol you should have this enzyme alcohol dehydrogenase because otherwise it is toxic, alcohol is toxic, so that is a problem. So, if you have this enzyme this will cut into other aldehyde or the ketones, right. Then also the catalyze in various processes like the oxidation and so on. Then this database called Brenda, right, because there are enzymes are wide variety of proteins with different functions, right. So, in order to give the importance of these enzymes, right, so they developed a database called Brenda. Brenda is the comprehensive enzyme information system, right. So, developed in Germany, right. So, it contains the information regarding different types of enzymes and different activities and the different functions and different properties, right. They started early in 2000s, right, and then they continued to collect the data and in between due to the financial crisis they stopped and then again they resumed to maintain the database and currently it is available, the Brenda is available online, right. So, it contains various information, right. For example, if you take any enzyme, right, first they have the nomenclature. So, there is enzyme names, enzyme number and the synonymous and so on, right, and they have the information regarding reaction and specificity, say pathways and substrates and so on and the functional parameters, right, the dissociation constant and IC50 values and pH and so on. Then it contains data on the isolation and preparation, how they prepare the enzymes and the stability, the structures and the different diseases and so on. Right. Also they give the regarding the applications and engineering. So, where this enzyme is applied because there are several enzymes they are applied in industries, right. So, all the information they give in the Brenda database. right. This is the website brendaenzymes.org. So, you can get the information from this website, fine. So, now when they form these enzymes, so database they give 4 different numbers, right. This is called enzyme commission number, and they have 4 numbers separated by dots. For example, here I mentioned EC 3.4.11.4, right. So, each number and each classification they have some meaning, right. For example, if you see this one EC 3.4.11.4, right. So, there are different types of enzyme. This mean for a specific type of enzyme. First one is the first class, right. So, the EC3, right, these are the hydrolyses, right, these are enzymes that use water to break up some other molecules, right. There are different types of enzyme classes, right, they are classified into 6 top level codes, right. So, EC1, EC2, EC3, EC4, EC5 and EC6. If we give this number, for example, if we do EC2, immediately we know that enzyme belongs to transferase. If it is EC1, it is oxidoreductase, right. So, if we do EC1, it is oxidoreductase, and you can see it catalyzes this oxidation or reduction reaction. Okay, these are the examples, right, oxidase and dehydrogenase. Here AH plus B is given as A plus BH, reduced or it is oxidized. And EC2, these are transferase, right, you can see the reaction AB plus B, this gives A plus BC. And EC3 is hydrolase, this involves this hydrolysis, right, AB plus H2O, you can see water molecule here, and here AOH plus B into H. And EC4 is lyase, and EC5 is isomerase, and EC6 ligase. Likewise, there are 6 top level classification of enzymes. Then each top level has subclasses. If you see here EC3 is, what is EC3? These enzymes are hydrolase, right, because they use water to break up some other molecules, right. This is EC3 is hydrolase. Then if 3.4, this means they are hydrolase and they act on peptide bonds. If the 4 means it is acting on peptide bonds. And here go to next level 3.4.11. So, E is the 3 is the hydrolase and 4 is in the peptide bonds, and 11 it gives the cleavage of the amino terminal of amino acid, right. And the last classification you here 3.4.11.4, these are hydrolase and they are acting on peptide bonds, and N terminal, right, the amino terminal of end of this tripeptide. So, this we give the number, right, then easily you can see the type of this specific enzyme, right. So, now you can access this database, Brenda database, and for example, I give information regarding EC1111, this alcohol dehydrogenase. So, here you give the data for all the alcohol dehydrogenase they deposited in Brenda database. They give the enzymes and they give the small molecules and they give the reaction and they give all the information regarding alcohol dehydrogenase. And in the particular case, if you take alcohol dehydrogenase, only few residues are acting as catalytic sites, right, that I discussed earlier. So, to understand the catalytic site residues, okay, there is another database called catalytic site atlas, right. This is CSA, catalytic site atlas. What they did? So, they collected the information regarding the catalytic site residues of enzymes, right. I discussed in the previous class about databases, right. So, the data are scattered in the literature, right, when they solve the structures, right, when the data are available in the literature, but here they collected the information regarding the catalytic sites of all enzymes. They got the information regarding enzymes, and from the enzyme they collected the catalytic sites and they developed this database called catalytic site atlas, right. And once we have sufficient number of data, then these data are also useful for identifying these residues if the sites are not known, right. So, in this case they used the data available in this database and to study the characteristic features and analyze what are the features important to identify the catalytic sites. Using the information, they developed some models to identify the catalytic site residues and they predicted the catalytic site residues for all the enzymes, right. If the structure is not known, they use the sequence information and identify the catalytic site residues. They included that information also in this database. So, it contains both the experimental data as well as the computationally identified catalytic site residues if structures are not known. So, now you can get the catalytic site information with based on various aspects, such as you can search the PDB codes. If you give PDB code here, you can search, and then you can give the switch prot code or the uniprot code here or the EC number, right. So, here I give the EC number 1111. What is 1111? Alcohol dehydrogenase, right, just I gave this case 1.1.1.1 is alcohol dehydrogenase. If you give this one, then it will show you the all the PDB codes related with that alcohol dehydrogenase. How we get so many PDB codes? Different organisms and different mutations and different resolution, right. So, there are various options to get the same proteins with different codes, right. So, here we can see the different codes, and if you select any of these codes, say 171, right. So, then here you can this is for 8 ADH, right, this is oxydoreductase, right, and they give you the number which is the functionally important residues such as catalytic site residues, right. Now, if you go here you can see some letters in red, right. So, the here, so you can see this one, this serine, this histidine and this leucine, right. Here these are the residues, right, which are mainly the catalytic site residues, right. So, I can put it here, right. So, the one which is shown in red, right, they are the catalytic site residues for this particular protein, right. But if you have any enzymes, if you like to know about the catalytic site residues, just you can use this CSA, right, to identify the residues which are behaving as a catalytic sites, right. Fine. So, now discussed about the enzymes, right. So, enzymes, what are the enzymes? Yeah, the specific proteins which catalyze reactions. The active, what is the active site? Reaction happens, there is a small part of the residues, 3 to 4 residues, right, that site which are having this reaction, that is called active sites, right. So, then we discussed about different types of databases. What is the database for the enzymes? Brenda database. And what is the data where we can get the catalytic site residues? Catalytic site atlas, right, we can get the information regarding enzymes.